I'm going to give you basically a talk on 2 mosquitoes, but I'd like to thank the CSIRO cluster for allowing us to build capacity in our lab. I normally do research on malaria in the Pacific, and we were able to move into looking at these 2 vectors of dengue and the newly emerging chikungunya virus. So we know, the planet is warming. This is just the mean temperature from 1970 till now, and we know the rainfall is changing, and this is very consistent with projections. So in Australia, there are certain parts of Australia that are drying, consistent with projections, and the northwestern part is getting a little more wet. Now, to deal with this drying, a lot of the state governments have been trying to drought-proof their urban regions by using rainwater tanks, and this has been a good thing. But there are certain trade-offs for that good thing, and rainwater tanks, for certain species and exotic species of mosquitoes, are fantastic reservoirs for larval development. They're large, they're well buffered, and they allow a large productivity of mosquito larvae. And our population, highly urbanised and in those regions that are drying, and the amount of rainwater tanks are growing. So I want to talk about really Australia's most pressing urban vector-borne disease problem. Dengue, which you probably all know, we have dengue epidemics, we've been having them yearly up in far north Queensland where the mosquito Aedes aegypti exists. It's a tropical mosquito, and I'll talk a bit about that in a moment. We have this newly emerging chikungunya virus that's appearing around the world. It's being driven by a mosquito called Aedes albopictus. Now, some interesting phenomena has happened in our region. We know that dengue vector Aedes aegypti transmits dengue, but a large outbreak of dengue just occurred in the Solomon Islands this year, driven by Aedes albopictus, which is normally a secondary vector. These vectors are exotic, and I'll talk a bit about them in a minute. So our project with the cluster was to work out where they are now. Where will they go in a changing climate? And then Scott Ritchie will do 2 talks after that talking about innovative vector control. This is a snapshot of active dengue around our planet right now from the CDC. Dengue is an increasing disease, and really 40% of the world population is at risk of it. We have just gone over some dengue activity this year. There's dengue occurring in New Caledonia and a large outbreak in the Solomon Islands that occurred this year. About 6,000 cases, 85%. In the capital city, Honiara, about 6 deaths. I think there's the last Ministry of Health report. Chikungunya is actually rocketing through New Guinea right now, being driven by the mosquito Aedes albopictus. So what I want to talk about first is Aedes aegypti. It's an exotic mosquito. It's come from Africa. It lays its eggs in tree holes, so relatively clean water. It traditionally would bite primates. It's highly domestic. It transferred to moving around with humans. Larvae develop in artificial containers, not in puddles, not in swamps. So humans have artificial containers in their domestic habitat, and these fill up with water, and that mosquito will breed very happily, feed on humans, and will lay their larvae in those artificial containers. The eggs are desiccation resistant, so when those containers dry out, they can be moved around, so populations do get moved around of this mosquito. It was historically extant through much of Australia last century, but over the last 60 years, because of the change of our water management, moving from domestic rainwater tank drinking water to reticulated water in the middle of last century, the mosquito's tropical biology could not permit it to exist outside of that biology. But we are now revisiting that because we are trying to drought-proof our urban regions. And we are resupplying our urban regions with rainwater tanks. So this is a map of the historical and contemporary distribution of Aedes aegypti. In red is the historical distribution. This is the distribution before 1980, and actually towards the middle of the last century. The shrinkage from this population and this population back up to more tropical Queensland correlated with the removal of domestic rainwater tanks. And this has been modelled by Mike Carney from Melbourne University. So we're at a stage now where we only really have the dengue vector in Queensland and one site in Tennant Creek in the Northern Territory. In the early part of last century, there were dengue epidemics that used to start in Queensland. And they would start in summer, peak in autumn, and they would stop in winter. And they would roll out into New South Wales, only stopping when the temperature got too cold for the virus transmission to go through the mosquito before the mosquito died of old age. That temperature was a 14.2 degree wet bulb temperature, TW, which has an aspect of humidity into it. And we can use that as an isotherm to get a sense of where transmission can occur or should be shut down on the Australian continent in winter, where you can get year-round transmission. Again, this is where transmission shuts down to a hypothetical value from observations, or where it can occur all year round. So this is a concern, as the vector is in our urban landscapes. International travellers often fly into our capital cities, infected with dengue. When infect the mosquito will cause an epidemic. And really the humans are the vectors here, not the mosquitoes. So we wanted to know and revisit these collection sites in Queensland and collect mosquitoes and do some population genetics on them. So we visited a lot of these sites, and this was a collaboration between University of Queensland, CSIRO, the Queensland Government and local authorities. And we found populations 250 kilometres away from Brisbane. In Roma and Charleville and some of the other regions. And Rockhampton had a very healthy population of Aedes aegypti. But we wanted to look in Queensland, I would look in Brisbane, because we're concerned this is an international airport, people flying in with dengue. If we have Aedes aegypti in Brisbane, it's a major problem. So as part of the class, we instigated a large survey, and this was run by QMR and local council. And it was quite an enormous survey. They looked at 56 statistical local areas, about 52% of Brisbane's SLAs, the little map here of what they had covered. From 5,000 houses successfully surveyed, and that's 40% of visited properties. There were a lot of denials, and often people weren't home. 400,000 mosquito larvae were collected, 70 species found, and over 40,000 containers were investigated. This is a huge data set. It's now being mined by the CSIRO. Luckily, no Aedes aegypti were found in Brisbane, but we found that garden accoutrements, your bird bath, your kids' toys, pot plants, buckets, were primary productivity areas for these domestic mosquitoes. Not just Aedes aegypti, but some of the other species could be used as proxies of where aegypti might go. In this survey, they found, by looking at over 2,000 rainwater tanks, and these are proper government subsidised rainwater tanks that were initially compliant. When installed, 4% were not compliant. Now, these are really hard to sample them quantitatively, because think of how a rainwater tank is and how do you get into them. So we're trying to develop techniques of being able to detect DNA in these rainwater tanks. But basically, 1 in 2 households in Brisbane owned a rainwater tank, and the spatial movement of Aedes aegypti through work we've been doing, looking at the spatial movement of this mosquito, is about a neighbourhood block. So one defective tank in a neighbourhood will supply enough mosquitoes for that whole block. And again, we're not even talking about in a warming climate with this human adaptation, will we see an expansion of Aedes aegypti beyond last century's distribution and into cities? Well, perhaps we will, but that for another day. So I'm going to move across to another mosquito, and probably a bit more of a problem. This is Aedes albopictus. It's the Asian tiger mosquito. It's a container inhabiting like Aedes aegypti, but it's polyphagous. It doesn't just bite humans. Aedes aegypti will mostly bite humans. This mosquito will bite your bird, your dog, your cat, most vertebrates, really. It's got some ecological plasticity, so even though it exists in the urban landscape, it can exist in the peri-urban and rural and forested landscape as well. So once it gets into an urban area and you try and eradicate it, it'll move into the sylvan landscape and it'll be impossible to eradicate. It transmits dengue, we've just found out, because of a big dengue outbreak in the Solomon Islands. I don't think it's quite over yet, I think it's still going on. Chikungunya, the emerging chikungunya around the world is occurring. The original outbreaks were in Reunion Island down here, but they're occurring worldwide, and there's a large outbreak occurring through Papua New Guinea right now. And probably what the public will dislike even more, it's a very big nuisance. Unlike Aedes aegypti, it's an aggressive biter. It's been given the name of a backyard barbecue stopper, and where it's invaded Italy and America, it's causing havoc to the alfresco lifestyle. So what facilitated this incursion? And we thought initially at the outset of this project that it was climate variability, and let me explain. So the native distribution of this mosquito is Southeast Asia. It got into West Papua or Papua about 1962, 1972, into PNG itself. We don't know when it got into Port Moresby, but surveys by the Australian Defence Force in 1992, and there were some surveys in 1998, found it basically on the island of Daru. It's a provincial capital of Western Province, and so international travel has to pass through Daru, probably how it got infested. But the surveys in 1992 did not find Aedes albopictus along what we call the southern Fly River region, the villages that occur around there. But in 1997, 1998, there was a large El Nino event and a huge drought through Papua New Guinea. This caused a lot of water stress to the villagers there. And AusAid came to the rescue and supplied funding and installed rainwater tanks and 220 litre containers equivalent to a 44-gallon drum throughout a lot of these villages. We hypothesised that this allowed for a large population build-up. Work was completed in 2002, population build-up, and then a population expansion into the Torres Strait Islands, which we are seeing at the moment. And that took 12 years between the population that we identified here and it exploding into the Torres Strait Islands. So really, the story wasn't complete. In 2009, it got into the southern Torres Strait Islands, and Queensland Health has been battling and suppressing the population from these islands because they're transport nodes to the mainland of Australia. Huge amount of movement, commercial and domestic, through these islands, probably pushing the mosquito population. But we don't want it on the mainland. At the moment, the insecticide-based suppression is working, but it's probably not sustainable because mosquitoes evolve. So we wanted to perform a population genetic study on this mosquito, just to confirm that it was a population that expanded out of New Guinea. So we collected mosquitoes from Moresby, Lae, Madang. We also had mosquitoes from East Timor, and we got mosquitoes from Jakarta and Java, just as an outgroup. And what we did is we developed 13 microsatellites. These are the same sort of genetic markers used in forensic testing. And what we found, and surprisingly again, this is the island of Daru that had a population in 2002, and we hypothesised that this population extended into the southern fly region and out into the Torres Strait. But surprisingly, these are the populations, Daru, PNG, Madang. This is a factorial correspondence analysis. Each dot represents an individual mosquito assessed for 13 microsatellites. Their proximity to each other is their genetic relatedness, seen in 3 dimensions, if you can sort of pull 3 dimensions out of it. So these are PNG population, Madang, Daru, Port Moresby and Kuyunga, which is 700 kilometres up the Fly River, where we found Aedes albopictus. Indonesia, Jakarta, this is the incursion population in the Torres Strait. The closest group was material from Timor-Leste. There wasn't a perfect fit, and we've got more material from Indonesia to try and find out where they've come from. But we have a new hypothesis now. We know that the Macassan have been visiting our coastline for hundreds of years, before any European settlement. Great sailors. And today, these Indonesian fishermen are known to have entered Australian waters, and they've illegally fished for shark fin. This activity peaked between 2005 and 2007, so it was occurring earlier as well. And we know from DAF that several hundred landings have occurred through this region where they've stored water, shark fin, fuel, and this is where the incursions would have occurred, whether in the Torres Straits or in the southern fly region. And apprehended these shark boats, if you look in their potable water, they have Aedes aegypti and Aedes albopictus. So we've got sort of a smoking gun on that, and we have a new hypothesis. So is it a if or when Aedes albopictus gets to Australia, and where will it go? Well, Matthew Hill and Haria Hoffman did some really nice modelling that's in revision in a journal. They used the populations that invaded America in the 1980s and that occurred through Southeast Asia, and they built both mechanistic and correlative models for those distributions, and they mapped them onto Australia. And interestingly, the current distribution, or the hypothetical distribution for where Aedes albopictus will go in Australia, is basically all the way down the eastern seaboard, driven a lot by rainfall, driven a lot by orographic rainfall at that. The green is the North American population. It's theoretically will move inland, but as I said earlier, this is a highly ecologically plastic mosquito, and I'd be surprised if it didn't extend further inland, basically because humans buffer their environment, they store water, and the implementation of rainwater tanks into these urban regions will allow this mosquito, which is a temperate mosquito, not a tropical mosquito, to exist way out of its ecological niche and use humans as buffers. So the modelling suggests that Aedes albopictus will exist in all states, and I just want to give you some take-home notes about this talk. Non-compliant domestic rainwater storage will increase population productivity and distribution. This will just normally happen as these tanks age and people start playing with them. A warming climate will also increase population productivity, distribution, and the virus transmission season, so that in theory it'll become warmer earlier and it'll stay warmer longer, so a virus transmission season will be much longer in a warming climate. It's a big pest in urban landscapes, and really it'll impair our lifestyle. And they'll point their fingers at us and say, you let them in. So what we have to do is keep albopictus off the mainland. We need new innovative control methods, and Scott will be talking about some of them. We need to keep Aedes aegypti out of our cities, because if there's a vector in our cities, we know that people fly in from overseas with dengue virus, and that can come and infect our local populations, and spark off an epidemic. Yes, all wheels of dengue and chikungunya in our cities. And with that, I just want to acknowledge this is a large team. I was lucky enough to do the presentation for this, but there are a lot of people involved in this work, especially the surveys, which were quite grueling and took a lot of time. So thank you.